Hi everyone, it's Dr. Biology here, and this video is on the kidney and osmoregulation. It's part of A-level biology revision. So if we look at the spec, this is in AQA. Now if you're doing other specs, this is more than likely in your current spec. So this is related to control of blood water potential. So I'm going to go through the structure and function of the kidneys, particularly the nephron, and then talk about the role of ADH in reabsorption of water. So first of all, let's look at the main structures. Obviously, you've got two kidneys. You've got both kidneys, they contain a cortex and a medulla. Within that are millions of nephrons, and those nephrons have a really good blood capillary network around them. So we're going to talk about how things are filtered out of the Bowman's capsule or the renal capsule, and how things are reabsorbed. So ultrafiltration is the first part that we're going to talk about, and the formation of the glomerular filtrate. So the glomerulus is the capillaries inside the Bowman's capsule or renal capsule, and you'll see there's two parts. So the blood is coming through the afferent arteriole through and then back through the narrower efferent arteriole. So small molecules and ions are filtered out of the blood into the renal capsule or the Bowman's capsule. It doesn't matter which one you use. And ultrafiltration takes place due to the pressure maintained by the efferent and afferent arterioles. So the key thing is, why is the pressure higher? Well, it's because the afferent is wider than the efferent arteriole. So you get a build-up of pressure inside the Bowman's capsule. So therefore, more of the fluid is going to be forced out of the basement membrane through what we call podocytes into the cavity here of the renal capsule, and eventually down into what we call the proximal convoluted tubule. So here's a little animation. So here we go. So you can see you've got the glomerulus, the Notter capillaries, the Bowman's capsule, and you've also got the afferent arteriole, which is wider than the efferent. Then you've got the endothelium cells of the capillary, so there's gaps in the capillary to allow small molecules through. And then you've got the epithelium of the renal capsule, and that's got podocytes on it, and you've also got a basement membrane. The arteriole is narrower than the afferent arteriole, so pressure rises as the blood enters the glomerulus. And ultrafiltration, it takes place at the barrier between the blood in the glomerulus and the filtrate in the renal capsule. So here we've got the endothelium cells of the blood capillary. So they're obviously very thin. They've got thousands of pores, and they do provide a barrier to cells, but not plasma membranes. You've also then got a basement membrane, which is made of collagen and glycoprotein. It allows water and small molecules through it. Proteins, though, are too large and they're repelled by the negative charges on the fibers. And then eventually you've got the epithelium of the renal capsule. These are known as podocytes, and they're made of cells which are modified for filtration, which we'll talk about in a minute. So podocytes, they are like foot-like extensions. You can see there, and they've got small slits in them. They wrap around the capillaries of the glomerulus and they interlink with neighbouring cells. They fit together loosely, so they leave what we call filtration slits. You don't necessarily need to know they're 25 nanometers wide, but they're pretty small slits. And then the filtered fluid passes through the filtration slits. Here we go. Here is a group of podocytes. So what you can see is that ions and small molecules, they're going to pass through this filter. So you will need to learn what those things are, and they are water, glucose, amino acids, vitamins and drugs, and also urea. Cells, platelets and plasma proteins, so proteins in the blood capillary, cannot pass through the filter and they remain in the capillary. Right, okay, so here is a quick question. So it shows a diagram showing the renal capsule, shows the glomerulus, and the filtrate produce. It says, apart from water and glucose, name 2 substances which will be present in the glomerular filtrate. So that will be urea, amino acids, because they're small enough, fatty acids, glycerol, ions, or even small proteins. So water and glucose definitely, and then probably I would say urea and ions. It then asks you for 2 marks, and it doesn't say 2 marks there. The glomerular filtration rate is the total volume of filtrate formed per minute. Explain the effect on the glomerular filtration rate of a large loss of blood from the body. Now, you will find these kinds of questions in any exam. They are quite applied, and they're testing your knowledge that the fact that the blood is under high pressure, therefore forcing out the small molecules into the glomerular filtrate. So blood pressure is obviously going to be decreased, therefore it will form less filtrate. So this talks about the 3 processes in the kidney, but we're interested in ultrafiltration in this section at the moment. So describe how ultrafiltration produces glomerular filtrate. So this is quite a large question. You can have any answers like this. So if the blood pressure is high, or it's under high blood pressure or hydrostatic pressure, small molecules, and I'll probably name some of them. They pass through the basement membrane first, and then they possibly will pass through they will need to pass through the capillaries. You need to mention the presence of pores or podocytes, and then what can't get through are proteins because they're too large to go through. Sometimes you'll come up with calculations, so here's a good calculation. So I'd like you to pause, and I'd like you to read it and answer it. So in 3, 2, 1. Okay, so we first of all know that 75 milligrams of creatinine is created in 1 hour. Okay, so and it tells you the concentration in the blood. It says calculate the rate at which the blood was filtered in centimeters cubed per minute. So you're going to have to convert it. So 75 divided by 0.01 will give you the rate per hour, and then you're going to need to divide by 60 to get it per minute. So in this case is 125. So selective reabsorption. So obviously small molecules are being passed through. We've already said things like glucose and water are going to pass through the capillaries into the filtrate. Now those are quite useful substances, so your body's not going to let them all go off into the urine. So therefore you've got to reabsorb some of those molecules. And that happens in the proximal convoluted tubule, which is the PCT. So the filtrate flows along the PCT, and then it travels through the loop of Henle to the distal convoluted tubule. So if I just show you, it's going to travel through here, right around the PCT, which is coiled with a big network of capillaries around it, and then down through the loop of Henle, okay, and then back up through what we call the ascending limb to the distal convoluted tubule. So we're going to talk about what happens in that section. So useful substances do leave the tubules of the nephrons, and they need to enter the capillary network. So what is filtered out of the blood into the PCT? Well, we've already said what kind of things were filtered out. So here we go: water, amino acids, glucose, urea, and inorganic ions. Now, looking at that list, you want things like water, glucose, and inorganic ions to be reabsorbed. You don't want urea to be reabsorbed, because that's the poisonous waste that you're trying to get rid of. So on the right here, I've got a diagram, and you can see it shows you PCT cells. And particularly it shows you that they have certain adaptations. They have microvilli, and also there's a large amount of active transport. So this links to your year 12 work probably on glucose co-transport mechanisms. And in here we've got loads and loads of mitochondria that allow active transport. So a key question might be related to how are PCT cells adapted for their function? Well, they have microvilli, so that increases the surface area for the reabsorption of useful materials from the glomerular filtrate, and they have mitochondria for active transport. So in terms of reabsorption, what's going to be reabsorbed is all the glucose. Okay, now if you don't reabsorb all the glucose, you're going to lose it in the urine. So that would suggest that you're unable to control the amounts of glucose in your blood. So you could be diabetic, so it could be used as a test for diabetes. And most of the water, depending on whether you're hydrated or dehydrated, will be reabsorbed, and some of the ions as well will be reabsorbed. So let's just look at some of the structures here. So we've already spoken about the loop of Henle, but if we look at the loop of Henle, you've got a descending limb, so it goes downwards, and when it goes downwards, water it's permeable to water, so water is lost, but mineral ions can still be absorbed into it. Okay, and you'll notice that it goes deeper down into the medulla, so where there's a high salt, low water concentration, and then when it's back up the ascending limb, it's actually then impermeable to water loss, but sodium ions can be removed. So we need to talk about how that affects the water potential in the loop of Henle, and how, as it says there, establishing a salt gradient in the medulla of the kidney, the reasons why that needs to be done. So we're going to talk about that now. So first of all, establishing a salt gradient. So first of all, the descending limb. So just a reminder, the descending limb is there, okay, and it's permeable to water. The ascending limb of the loop is permeable to salts, but not water. That's the ascending limb. So this means that as the loop descends into the medulla, the interstitial fluid becomes more salty and hypertonic. So outside the loop of Henle here, it's getting more and more, the salt concentrations are increasing. So additionally, the blood network that surrounds the loop of Henle flows in the opposite direction. So for example, if I was to draw a really bad capillary network here, rather than going downwards, it's going back, actually back up this way against the concentration gradient. So let's look at that in a slightly more detail. Okay, so here we've got the loop of Henle. So as the fluid in the tubule descends down the descending loop, the water potential becomes lower or more negative, okay, because water is being lost by osmosis. So the walls of the descending limb are permeable to water only. Well, not only, sorry, the salts can actually enter. Water leaves by osmosis into the surrounding tissue fluid. So sodium and chloride ions can then diffuse into the tubule from the surrounding tissue fluid. Secondly, on the ascending limb, so then it ascends back up towards the cortex of the kidney, so the water potential now becomes higher or less negative. The reason being is due to the fact that sodium and chloride ions are diffusing out of the tubule into the tissue fluid at the base. So higher up the tubule, sodium and chloride ions are being actively transported out. So this is to raise the water potential as you reach the distal convoluted tubule. So the walls of the ascending limb are impermeable to water, so that cannot leave the tubule. Then we come to this thing called a hairpin countercurrent multiplier system. So the fact is you've got two different directions, and in one direction water is being lost, and in another direction sodium and chloride ions are being lost. So water moves out, as we know, and it makes the fluid in the tubule very salty. So then salt diffuses out of the base of the ascending limb. Because of all the salt, it's changed the water, it's changed the concentration gradients, and it means that as it's at a high concentration, so it's very negative water potential. And then it's transported out using active transport at the top of the ascending limb. So the removal of the ions from the ascending limb makes the urine very dilute, and therefore water can then be reabsorbed by the body from the distal tubes and the collecting duct. So here we've got the distal convoluted tubule. Okay, so the tubule fluid passes through it, and active transport adjusts the concentration of various salts in the tubule. So now there's a larger amount of water, so there's a high water potential. So we get to the collecting duct, and the fluid reaches it, and it has a high water potential, so it's less negative. And then it carries fluid. The collecting duct carries the fluid back into the medulla, which contains a lot of salts, which is low and very negative, has a low and very negative water potential. So the reason for that is to reabsorb as much water as possible. So as the fluid passes through, water moves by osmosis into the surrounding tissue, and then it's able to enter blood capillaries, and then it's carried away. So water has now successfully been reabsorbed. Yes, there's a water droplet. Okay, so that kind of explains how water is reabsorbed and the importance of sodium ion gradients. Let's talk a bit about the role of ADH. Okay, now this is quite easy really to remember. If you can remember what happens when ADH levels rise, it's basically the opposite for when it decreases. So the first thing is ADH is a hormone. It's released by the posterior pituitary gland, but several things need to happen before it's released. So first of all, let's imagine you are dehydrated. So when you're dehydrated, blood ADH levels will rise. Okay, and we need to talk about why they rise. Well, when you're dehydrated, your water content in your blood drops, so its water potential drops. So this is detected by osmoreceptors in your hypothalamus in your brain. So the posterior pituitary gland then releases more ADH. Okay, then it travels through the blood to the DCT and collecting duct, and therefore making them more permeable. So it's making those pores more permeable and wider, so it allows more water to be reabsorbed back into the blood through the capillary network. So if we look at the opposite, okay, I'll move myself, there we go. So in this case, blood ADH levels fall when you are hydrated. Well, why? Because the water content in the blood rises, so its water potential rises. So this is detected by exactly the same osmoreceptors in the hypothalamus, and therefore the posterior pituitary gland releases less ADH. So therefore the DCT and collecting duct become less permeable, so those pores get smaller or they close, so less water can be reabsorbed into the blood by osmosis. So it's as simple as that really. You don't have to go into great detail about how the pores are permeable, what happens in the pores. You just need to be able to pretty much say what I've just put. Okay, let's have a look at some exam questions. So what I suggest you do whilst we go through these is to probably pause, read them and have a go yourself, then we'll go through them. So pause in 3, 2, 1. Okay, so this is a big 6-mark question. So humans can produce urine which is more concentrated than their blood plasma, so they're more concentrated. There we go. So explain, so the key command word is explain the role of the loop of Henle in absorption of water from the filtrate. So kind of answers you can have are the following. So let's talk about the loop of Henle. Now in this question they've started with the ascending limb. You could start with the descending limb. You don't have to do the ascending limb. So all those kind of key points. You do need to talk about water potential. So in terms of low water potential, high water potential. You could mention the longer the loop, the kind of lower the water potential in the medulla is. You need to talk about water leaving the collecting duct or the DCT. And because it doesn't say it in the stem of the question, you can say by osmosis. Okay, this question's a bit more kind of applied, so this is a very popular question. So I'd like you to pause it in 3, 2, 1. Okay, so it shows the concentration of the filtrate in different parts of one kidney. So the concentration. So it does tell you in the PCT, 99% of biological molecules are reabsorbed. Despite this, the concentration of fluid remains constant. So explain why. Okay, well, much of it is reabsorbed from the filtrate. Okay, so however, you could also say, well, water is also reabsorbed at the PCT, so it therefore will remain constant. So the concentration doesn't necessarily increase or decrease. Explain the shape of the curve in the loop of Henle. So it goes up and then down, so you've got to talk about both. So we can look at that in terms of concentration rises in the descending limb because the sodium ions enter and water is lost. Concentration falls because the sodium ions are then actively removed. But you could say water remains because its walls are impermeable to water. There you go. Here is about ADH. So again, I'd like you to pause and read and answer in 3, 2, 1. Okay, so what is the evidence in the graph that this person was secreting ADH? Okay, well, we need to talk about the DCT and the collecting duct, particularly the collecting duct, and you're looking for anything related to concentration rises because it loses water by osmosis. Okay, so it's increasing in concentration because water is being reabsorbed. So we need to say something about ADH. Well, it increases the permeability of walls for the collecting duct to water. Here's another one in terms of looking at data. So again, I'd like you to answer in 3, 2, 1. So the graph shows changes in the amounts of water, glucose and sodium ions as fluid passes along a kidney tubule from the renal capsule to the collecting duct. So first question, which hormone causes the decrease in water content? Well, there's only one hormone and that's pretty simple, it's ADH. This isn't though. Explain the change in the amount of glucose. So you can see it decreases greatly in the PCT, 100% is reabsorbed. Why? Well, reabsorption passes back into the tissue fluid. By what process though? And the process is active transport. Okay, so last one, explain the shape of the curve for sodium ions in the loop of Henle. Okay. There we go. So, first thing is sodium ions are pumped out of the ascending limb. So water passes out of the descending limb as well. And some sodium ions can re-enter the descending loop or limb. So high concentrations at the base of the loop. So some ions may diffuse out near the base, increasing the concentration outside the loop. Okay, so that brings us to the end. I hope you've found it useful. Please do subscribe and I will see you soon.